This presentation gives a brief introduction to the idea of polygenic schools, which is an important new development in genomics research. Education attainment, or EA, measures the number of years a participant has spent in full-time education. It's useful because it is collected in many large-scale studies and is a readily available metric. Unlike measures of intelligence like IQ or G, it has been suggested that it is unbiased in the sense that they are not subject to training, faking or anxiety, each of which is a criticism of intelligence tests like WAIS-4, which estimates G. This is a Manhattan plot for a GWAS study of 1.1 million people for educational attainment. The paper identified differences in over 1,200 variants that were statistically significant. These were distributed evenly across the whole genome. The DNA variants in this analysis account for about 11% of the differences or the variance between the individuals in the study for educational attainment. This is remarkable, but it is still some way below the 50% or so that we would predict is the genetic contribution to the differences between people for educational attainment. We expect that if we used a larger sample of people and a screening of the whole genome, then we would probably find the missing variants. The remaining 50% of the differences would be explained by differences caused by environmental factors, reinforcing the key idea that phenotypes like educational attainment arise through interactions between genomes and the environment. Not all of the variants are in or are near protein coding regions of the DNA, and yet they are statistically significant in this analysis. Their functions will doubtless be discovered in the future. However, many variants are associated with nearly 2,000 protein coding genes, and these variants are located in or close to regions of DNA that are expressed during fetal development of the nervous system and also in childhood and adolescence. Many of these variants are associated with genes that encode for proteins that produce nerve cells or neurotransmitters, or activate ion channels, or promote cell signaling or synaptic plasticity. In other words, these are the variants that help to build and maintain brains. GWAS studies of educational attainment show a significant genetic component. As we've seen, the latest version, EA3, predicts about 11% of the differences in educational attainment. This is important because it's now a usable characteristic, rivaling socio-economic status in importance as a research tool. For example, parental EA correlates 0.3 with children's intelligence, accounting 9% of the differences between children. So, how do we calculate a polygenic score for educational attainment? Well, imagine that a GWAS study on an individual finds 10 single nucleotide polymorphisms or 10 variants as shown on the screen. The first step is to work out which of the two alleles has the largest effect on the phenotype. For SNP number 8, allele A has the increased effect on the phenotype, and as there are 2 copies of allele A present, the combined contribution of these alleles is 2. That is the genotypic score for that SNP. For SNP5, it is the allele G that has the increasing effect on educational attainment, and in this particular case, the person does not have a copy of G, so their genotypic score is 0. When one copy of the increasing allele is present, the genotypic score is 1. This is the case for SNPs 1, 3, 4 and so on. Added together, we get a total polygenic score of 9. However, this is weighted by looking at the correlation of each SNP to the phenotype, and then this is used to produce a weighted genotypic score by multiplying the correlation with the genotypic score for each allele. It is clear from these analyses that the effects of any one individual variant is very small. The SNP with the largest effect accounts for only 0.02% of the differences in EA, which is equivalent to about 2 months of education. Polygenic scores for EA are obtained by considering the effects of all the variants in the analysis, even those with low levels of statistical significance. Their effects are aggregated into a single polygenic score, which is called a GPS. Twin and adoption studies predict that about 50% of the differences in IQ scores in a population are due to genetic differences. EA3 predicts only about 10% of the differences in cognitive performance. This difference, which is found in GWAS studies of other characteristics too, has the general term missing heritability. This paper, The New Genetics of Intelligence by Robert Plomin and Sophie von Stum, was originally published in Nature. It is a significant paper because it introduced the idea of polygenic scores to a wider audience beyond the genetics community. It's very readable and very accessible. I suggest that you read it and talk to each other about it.